Hello viewers, in this particular lecture in microbiology, I shall be discussing the different aspects of bacterial sexual reproduction. As you know, bacteria is a group of prokaryotic organisms which do not have true nucleus. So at the same time, they do not have proper demarcation of their life cycle. That is, the haploid and the diploid phases are not properly demarcated. And they have only some DNA which can be in the nucleoid or some extranuclear DNA. So the sexual reproduction in bacteria is not like other organisms or other eukaryotic organisms. The sexual reproduction mainly can be of three major types, which are conjugation, transformation and transduction. There is a fourth type which is called lytic or lysogenic cycle, which is mainly the mechanism for reproduction in virus, but we also consider it under bacteria because sometimes when a particular bacterial virus or a bacteriophage which is attacking the bacteria will take up some bacterial DNA and will take it to another bacterial cell. So thereby we can say that the bacterial DNA is perpetuating from one cell to other. So as I was telling, you can see that bacterial reproduction or bacterial sexual reproduction can be of three major types, that is conjugation, transformation and transduction. You can see here in this figure, conjugation maintains a clear contact between two bacterial cells. Transformation is a DNA which is present outside the bacterial cell, maybe in another bacterial cell, or maybe as a naked DNA outside, will get into the bacterial cell and modify it. And transformation is followed by transduction, where there will be a bacteriophage involved. The phage particle actually invade the bacterial cell and will put in the DNA inside, and thereby when that particular phage particle which will come out after lysis of that particular host cell, they will contain some bacterial DNA and that will perpetuate. Now coming to the first process, which is conjugation. It was discovered by Lederberg and Tatum in 1946. In this process, what happens is two bacterial cells will come close to each other. They form a conjugation bridge and thereby they will exchange the genetic material. Now this genetic material is naturally not in the nucleoid. It is present as an extranuclear DNA or a plasmid, which is exchanged after replication. So they discovered this particular process in a unique experiment. And what was that experiment? Again, you can see in this figure, there are two bacterial strains. They were mixed, and this particular bacterial strain, strain A, was unable to synthesize some of the amino acids like methionine or biotin, and similarly, the strain B was unable to synthesize threonine or leucine or thiamine. Now, when they were mixed, these two particular bacterial cells were mixed. What happened was they developed some blue colonies. You can see in this figure the blue colonies actually indicate that these particular colonies can produce all the amino acids. So they started with a particular two strain where all the amino acids were not present, but ultimately they got back some individual strain which could produce all the amino acids. So naturally there has been some transfer of genetic material from one to other, enabling the new colonies to produce all the amino acids. Now this method involves the actual contact between two bacteria, which was explained by Bernard Davis. And Davis, what he did was, he did a unique experiment which is called the U-tube experiment. What he did, you can see, there is a U-tube where the strain A and the strain B were separated by a fine filter which actually prevented from the free mixing or intermingling of the two bacterial cells. So when it was done, one bacteria was not coming in contact with the other. So when he did it, and then when he tested for the production of the amino acid, he found out that none of those bacteria could produce all these amino acids. In other words, conjugation failed. So thereby it was proved that conjugation involves direct physical contact. Now coming to the most important phenomenon, I told you that a bacterial cell is having a nucleoid and there are also external plasmids or external circular DNA. Now these plasmids will have some factor which commonly is called the F factor or the fertility factor. Now this F factor was denoted by William Hess in 1953, and this F factor or the fertility factor is present in some, not in others. So where it is present, it was called the F plus, and where it is absent, it was called F minus. And this F factor, he actually described it and he called it F plasmid. You can see in this particular figure, there are several specific areas of the F plasmid where there is the origin of replication, the tra region, the insertion region, and also the modification region. So this particular plasmid ultimately has to be subject to transfer from the donor to the recipient. Now we come to the mechanism of conjugation in bacterial cell. Now what happens is, you can see there is a donor and there is a recipient. The donor is having this extranuclear circular plasmid, and this plasmid is actually producing a strand, and this strand is entering through this conjugation bridge into the recipient. Perhaps this figure will give you a better idea of how conjugation occurs. You can see that there are two different cells. One cell is the donor, which is having the F plus factor in red, you can see, and there is the F minus, which is without this red, and then there will be conjugation bridge. Essentially it is formed of pili, and this particular conjugation bridge will bring the two bacterial cell closer. And then what happens is, I showed you that origin of replication at the ori T, they will start the replication process with a nick, and after this nick, gradually this strand will open up on the other side, and thereby the DNA, the daughter DNA will be produced. And then what happens is, at a particular time, this particular strand is going into the next recipient, and this strand is remaining into the donor, and the two strands now are still joined, but ultimately they get separated. So ultimately there should be a second nick appearing. After the first nick, actually the strand started replication. The second nick will actually terminate the replication process, and afterward this particular F plus has actually modified the F minus into F plus. So this process of conjugation is denoted between the F plus and the F minus strain, where F plus is modifying F minus to F plus. Okay. So there is another unique process. I have told you that there is F plus which is modifying F minus to F plus. Now there is definitely the F plus getting into the F minus. It so happens, you can see in this figure, the F factor which is received by the recipient can get into the nuclear genome or nucleoid of the recipient, and they will produce a normal strain which is not present originally, and it was called HFR strain. Now this complete transfer may require about 100 minutes. After that, they found out, it was Sforza in 1952, he found out there is another strain where there are some properties present like that of the F plus particle, but this property is different in the sense there is no external F plus plasmid. So naturally this strain was called high frequency recombinant strain, and it was called the HFR strain. So this particular HFR strain formation, you can very well understand, is the transfer of the F plus genome and then entry or the application of this nick in the parent genome of the recipient, and then infusion of the particular recombinant DNA into the nucleoid. So this phenomenon will modify an F minus to F plus, and then F plus getting into the nucleoid will produce the HFR strain. So basically this is a recombinant type of HFR genome where you can see specific amino acid producing region, arginine region, serine region, histidine region, tryptophan region, and there is also the F factor. So this is more or less that particular HFR genome of E. coli. It is also circular in nature. But what we actually have learnt is that due to conjugation, we can have a phenomenon where there will be F plus in the plasmid, and the complete genome is called the A plus. So F plus A plus, whenever it is combining with F minus A minus, will produce another F plus A plus, because obviously that genome is getting transferred. But sometimes it so happens that F plus A plus will actually convert a particular strain into HFR. That means that plasmid which is getting in will get into the nucleoid. So HFR strain which is actually attacking another F minus A minus will produce F minus A plus or F minus A minus. So always what I am trying to say is that HFR strain may not produce productive conjugate or productive conjugated bacterial cell. Right? You understood this. So you can see that HFR strain can definitely undergo conjugation with another HFR strain, but it will be a non-productive conjugation where there will be no transformation noted after, because that particular nuclear genome will be modified but not the F factor, because it is producing an illegitimate conjugation. As you can see that plasmid, apart from that F factor, is also having other genes. So there was another unique genes or set of genes discovered in this plasmid, which was the resistant plasmid or R plasmid. Now this resistant factor is basically resistant to antibiotics. So bacteria are resistant to antibiotics, and these genes are present within that particular bacterial plasmid. And these R plasmids, they have seen that, say, Shigella, which is a gram-negative pathogenic bacteria, is actually transferring the R plasmid, and as a result of which the recipient becomes resistant to different set of antibiotics like the ampicillin, the tetracycline, the kanamycin, the streptomycin, the sulfonamides. You can see they are represented as CANAR, SMR, SUR, AMPAR, AGR, etc. And AGR, as you know, is another unique point. AGR is hydrogerm or mercury. So they are resistant to mercury. That means they can survive in a particular environment containing mercury. So this particular transformation by the entry of this R plasmid will make the bacteria resistant to antibiotics. And as a matter of fact, there are different such characteristics present in the plasmid, which is denoted in this table. You can understand the fertility factor or the resistant factor is F or Ri. The bacteriocin is denoted in E1. Bacteriocin, as you know, is a protein which will prevent the growth of other bacteria. The heavy metal resistance in R6, the enterotoxin reproduction, enterotoxin is a toxin producing ability in ENT, or the camphor metabolizing ability in CAM, and the tumorigenicity in Agrobacterium tumefaciens in the TI plasmid. And that reminds me that a particular generic form, say Agrobacterium tumefaciens and Agrobacterium rhizogenes, they can be transformed, they can change themselves by taking up that plasmid from one donor to recipient and vice versa. So this type of intergeneric transfer is very common in Agrobacterium, which will modify their character, and at the same time it is also noted in E. coli and a totally different genus of Streptococcus. So what happens is a particular, say, atar locus has been transferred into E. coli, making the E. coli plasmid transformed, and thereby one particular bacterial character is getting into another bacteria, modifying its character. So if we now come to the actual application of conjugation, what we see? We see, number one, bacteria started as a group of organisms, but that group of organisms is getting modified and producing new set of organisms. So this is one, that is evolution of bacteria. And now, you know, human always try to get some opportunity to utilize it. So that plasmid, they understand, okay, they are very good vectors. They can be used as a vehicle to transfer gene. So this particular mechanism was used in the transfer of gene. So these plasmids were very good tools for genetic engineering. And most importantly, obviously this is not a good point though, but bacteria is gradually becoming resistant to antibiotic, which is actually making our life more difficult, because the more new molecules we discover, the bacteria gets one step ahead and become resistant to that particular antibiotic. We come to the next important mechanism of bacterial reproduction, which is transformation. What is transformation? Transformation is a phenomenon by which a DNA fragment which is present in some other bacteria or maybe in some other organism which will enter into the bacterial cell, resulting in its modification. Now it was a historical experiment done by Frederick Griffith. He actually did an experiment with mice. He explained the phenomenon of transformation but couldn't explain the mechanism. So what he did was, he took this pneumococci. Now this pneumococci bacterium existed in two separate forms. One is the type 2R, which is rough type because of the pili, and another one is the type 3S, which is smooth type because of the presence of the external glycocalyx layer. So initially it was observed that when 2R was injected in the rat was surviving, so it was called that the 2R was avirulent. But when that particular smooth, you can see this smooth bacteria was injected into the rat, it died because of pneumonia. So that means that that particular smooth type is virulent type. So definitely this is causing the pneumonia disease and the rat dies. But then what happened was, he heat killed it. By heat killing, he actually removed the external wall and the bacteria became dead or couldn't cause any infection. So when this heat killed strain was injected, then they definitely found out that the rat was alive. Now then he did a unique experiment. He introduced the normal 2R and the 3S strain together, which is the 3S here is heat killed. So technically the rat should have been alive because the 3S was dead and the 2R was avirulent. But what happened was the rat died, and there actually he called it Griffith's effect. But this particular explanation of transformation, here is definite transformation, that is 2R is getting modified to active 3S by the presence of the DNA of the 3S debris or the heat killed 3S. So this phenomenon was explained by these three scientists. It was in 1944, much later, by Avery, MacLeod and McCarthy. And they did a unique experiment. What did they do? They took this 3S type, and then in one particular case there was no protein because they treated with proteinase. Another one was ribonuclease, and the other one was no DNA because they treated with deoxyribonuclease. Now what happened was, when this R was added in both the cases, you ultimately get S in the first two. But in the third one, where there was no DNA because it was degraded using DNase, they didn't get any virulent S cells. So here this transformation of R to S was ultimately brought about by DNA present in that particular bacterial cell. So thereby two things were explained. One was that DNA is the genetic material even in particular bacteria and also in viruses, but this particular DNA is also helping in transformation. So both transformation as well as DNA as a genetic material was explained by this unique experiment. Bacteria is not only modifying another bacterial cell, but sometimes bacteriophage can also modify bacterial cell. You can see in this particular slide where the M13 phage is attacking a particular E. coli, and it is definitely received by specific set of P3 protein. And when this P3 protein is present, there will be successful binding of the phage with the E. coli, and this particular E. coli will definitely produce more P3, and naturally they become more prone to infection by bacteriophage. And in those cases where the P3 will not be there, those E. coli will be resistant to the bacteriophage. So what we can see is that a particular E. coli is getting more prone to infection with the production of a protein which originally have originated from a bacteriophage. So that is also a case of transformation. And this particular transformation definitely means that the cell has to accept external DNA. So that phenomenon of accepting external DNA is known as competence. Now competence can be natural. You can see a simple bacterial cell is there, a particular channel is formed, and this particular channel is getting rid of bacterial chromosome. It is getting accepted by another bacterial cell, and the second bacterial cell getting transformed. So this phenomenon is called natural competence. And in natural competence, what they have shown is some definite protein involved. The protein secretin or the protein secretory system. And there is also another important type 4 pili, which is actually involved in the translocation of the particular genome or the particular gene fragment by translocase, and definitely this translocation is dependent on a process called the rec-A process. So definitely here pilin is having some role in the transfer of the gene fragment from one bacteria to the other to induce natural competence.
Sir, the pili plays such a prominent role here. What is it made up of?
Yeah, you see, definitely the pili is an important constituent of a bacterial cell. The pili is formed of protein, and this protein is pilin protein, pilin A or pilin B, and you know this pilin protein can undergo degradation. Whenever it is undergoing degradation, the pili length becomes small. So when the length is becoming small, they are bringing in two bacterial cells close to each other, which is in conjugation. But in case of transformation, what you see is the pilin is present as a specific receptor protein, which is allowing the DNA fragment to enter into the bacterial cell, because ultimately the production of pilin is because of the secretin protein. So one is acting as a conjugation tube formation, and the other one is it is acting as a receptor protein. Bacterial cells can be taking up the DNA. We can induce that competence. Now this competence is called artificial competence. Artificial competence can be of different types. It can be definitely, one is heat shock. You can see in this particular E. coli strain where they are subjected to heat to 42 degrees centigrade. So by this temperature treatment, the cell wall becomes definitely sort of permeable to that particular gene fragment. So when they are treated to temperature and when they are plated, they could grow in presence of ampicillin. So here what we can see is by temperature treatment, the cells have become actually permeable to particular transformation. Similarly, another important phenomenon is application of electric charge. Now electric charge, whenever it is applied, there will be definite negative and positive charges created outside and or rather inside and outside of the bacterial cell, and then there will be some smaller channels created because of this positive charge, and thereby these channels are utilized for the entry of the DNA fragments. So electric pulse is one of them, and heat shock is another point to induce artificial competence. Perhaps the concentration of calcium is also a very important phenomenon in the application of inducing competence to a bacterial cell.
So what is the role of calcium chloride in inducing transformation?
Yeah, calcium chloride is applied to a particular bacterial cell, and that particular calcium chloride makes the wall more hard. So naturally, it will allow the entry of the particular DNA fragment, maybe from virus, maybe from bacteria, maybe outside DNA, without causing any disruption into the bacterial cell. So definitely calcium chloride, if it is naturally present, it will induce natural competence, but if it is induced from outside, it will induce artificial competence. So what I was telling you, that artificial competence can be introduced by electric current or electric pulse. So this is a unique electroporation unit where you can see there is a particular cell suspension with electrode, the electric pulse is given, and this electric pulse is ultimately helping in the transformation of plant cells, and this transformation of plant cells will ultimately be brought about by this gene gun, where the gene is fired and this will enter into the plant tissue culture, and the gene will be actually attached to some gold particle, nanoparticle, and ultimately it will settle down and the DNA will get into the culture and will modify it. Now what type of DNA is commonly used in this process? In very common way, the DNA fragment which is used in modifying a particular plant tissue culture is the TI plasmid. This TI plasmid, as I told you, is actually obtained from Agrobacterium tumefaciens, which is modifying the plant cell. And you can see this circular TI plasmid is having the T region where there is the left and the right TNA border, and there is the opine catabolism and opine production gene, and there is also the virulent gene. Now normally this particular TI plasmid is inducing a disease called crown gall tumor, but this TI plasmid in the same way can be used in transforming the plant cell. How it is done? You can see this is Agrobacterium tumefaciens bacteria, which is producing the tDNA, and then the insertion of gene into the plasmid using restriction enzyme. Though we have actually shown the restriction enzyme, you can also use the same gene gun technique to transfer that gene. Then this is the modified the chimeric DNA fragment, and the chimeric DNA fragment is allowed to enter into the plant cell, and this particular plant cell is capable of producing that particular TI plasmid, and thereby there will be a modification in the plant cell which will be tumorigenic change in that particular plant cell, or otherwise if there is a particular gene fragment which is entered into the TI plasmid, this gene will express into the plant cell and will modify the plant cell. So in this way, using the TI plasmid vector, we can transform not only prokaryotic cell but also eukaryotic cell. Is it clear? Now coming to the last important aspect, which is the transformation of yeast cell. Yeast is also belonging to eukaryote. So here also you can see you are taking yeast cell, you are making them competent. Artificial competence is induced, and then there is a vector DNA which is allowed to enter, and you know yeast is quickly replicating by fission or budding, and that particular property whichever is present in the foreign DNA is introduced into the yeast, and thereby they are getting transformed. But ideally, in order to make the yeast cell permeable to external DNA, they are sometimes using glass beads. So in this particular lecture, what we have learnt is the sexual reproduction in bacteria. In bacterial sexual reproduction, we have conjugation, transformation, transduction, lytic and lysogeny. But here we have learnt mainly conjugation and transformation. In conjugation, there is a donor and a recipient bacterial cell, and the DNA fragment enters into the conjugation tube, transforming the recipient cell. And there is also another phenomenon of transformation, where there is an external DNA which is accepted by the bacterial cell. This DNA can come from another bacteria, from any other organism, but ultimately it will enter into the bacterial cell, causing it to change. But this phenomenon is utilized to modify higher organisms like higher plants or yeast. Thank you.